Okay. Thank you so much for coming to our fourth webinar. We have anticipated around 268 registrants from 46 countries. This is our fourth webinar about Gene Drive, and the title of the webinar is Regulatory and Governance Considerations for Gene Drive Research. I am Dr. Rodora Romero Aldemida, the Director of ISAAA Southeast Asia Center and the Global Knowledge Center on Crop Biotechnology. I'm a trained scientist with a doctorate in biotechnology and plant physiology from Purdue University, USA, and postdoctoral fellowship from Albert Ludwigs University, Freiburg, Germany, on beta-carotene enriched rice. So this webinar is hosted by our ISAAA Southeast Asia Center based in the Philippines in partnership with Outreach Network for Gene Drive Research. We gratefully acknowledge the support of our ISAAA Biotech Information Center network in Malaysia, China, Indonesia, Japan, Pakistan, Karachi, and Lahore chapters, Vietnam, Circa Philippines, and Thailand. The Gene Drive webinar series aims to help promote a productive and balanced conversation on the benefits and risks of possible gene drive applications, providing factual and accurate information that can help place the discussion under CBD in context. At this webinar, the fourth one, our resource persons will introduce the regulatory and governance frameworks within which research on gene drive takes place. It will examine what guidance materials are available to ensure that research is conducted safely, ethically, and responsibly. It will also look at the role of public consultation and stakeholder engagement plays in shaping research pathways and practices. So this webinar will run for 1.5 hours. We are currently live on stream at the ISAAA Facebook account and YouTube channel. All microphones are on mute and questions should be entered in the Q&A function only. The chat function will be used by the panelists and for important messages to the participants. Thank you all participants for responding to our pre-webinar surveys. And at the end of the webinar, we will provide a link to a post-webinar survey and respondents will be given certificates. So to start, let me now introduce our moderator, Dr. Viva Ahuja. She's the Chief General Manager of the Biotech Consortium India Limited. She has more than 20 years of experience in the area of biotechnology and biosafety capacity building. These include undertaking planning and implementing capacity building activities, imparting training through seminars, workshops, national and international training programs, and preparing well-researched publications for various stakeholders related to biosafety issues. She has been actively engaged in providing support to regulatory authorities in India, has expertise in the development of guidelines and also implementation of regulatory systems, including processing of applications and others. She has wide experience in issues related to the Cartagena Protocol on biosafety and is well-versed with relevant decisions and developments. Let us all welcome our esteemed moderator, Dr. Viva Ahuja.
Thank you so much for introducing the webinar objective to the audience. And at the outset, I wish to thank the ISAAA and the Outreach Network for Gene Drive Research for inviting me to moderate this webinar on a very important topic, which is the regulatory issues on gene drives. You see that as the audience would all agree, the regulation is the key to introducing any new technology. We need to be very sure that the products and technologies are safe for the users and the society, and at the same time are not, these are not over-regulated so that benefits can be harnessed by the society. Gene drives is a very exciting technology. You've had 3 webinars understanding about what are the gene drives and the applications, and today we are going to have the discussion on the regulatory issues. I have very, very esteemed speakers this morning with us, who will be speaking on these issues. the real experts on these issues. And I would like to briefly introduce the three speakers to all of you. First of all, we have Dr. Florida Carino, who is a biochemistry professor at the University of the Philippines and a member of the Department of Science and Technology Biosafety Committee. Dr. Carino has been involved in drafting policy and regulations for GMOs since 1994 and served in several training programs for international organizations involved in biosafety capacity building and for various Philippines government institutions involved in the regulation of GMOs. I also wish to add that, Florida has been very actively engaged in the negotiations at the CBD and CPB and has been a very, very useful, contributions to make. Our second speaker is Dr. Andy Shepard, who is a senior principal research scientist with CSIRO. He leads and he has led international collaborative research on ecological management of national priority weeds and pests in Australia, principally using biological control. His primary research interests are in the field of invasion biology. Our third speaker would be Naima Sykes, the most important topic of stakeholder engagement. And she is the stakeholder engagement manager at Target Malaria with over 10 years of experience working on stakeholder engagement and strategic communications with the government of Tanzania and multinational organizations operating across multiple sectors, including agribusiness, infrastructure, and extractives. Without wasting any further time, I would just like to go on to start with our first presentation and learn from Dr. Florida Carino on gene drives and issues on global governance. Over to you, Dr. Florida. You can kindly share your slides.
Yes, thank you very much, Viva, for your kind introduction. I will now see if I can get my slides going. I hope I have shared it well enough. I will be looking at gene drives and the various issues of governance and perhaps look at the different options that are available for us to refer to when we want to look at the governance of this particular development in technology. First, a disclaimer, the views expressed in this presentation are all my own and would not necessarily reflect any official position of the government nor of the DOST Biosafety Committee. That said, I can now go into the outline of my presentation. First, I'd like to have a brief discussion on the rationale for governance. And they include the landscape of governance mechanisms that will cover the entire range of work with gene drives, let's say from the research end up to the field releases, up to perhaps eventual deployment on a larger scale basis. And then look at a few of the attributes of an ideal governance framework for this new technology. Well, gene drives have been described before, but perhaps it is best to emphasize that gene drives are actually, natural gene drives are ubiquitous in nature. Although for now, we will limit our discussions on the synthetic self-propagating gene drive, and classically that would include the cassette inserted that includes a CRISPR-Cas9 plus whatever desired change you wish to include into the animal, plus the guide RNA that is being inserted into a reproductive cell. So we've had a discussion on this in earlier webinars. Why look for governance of this new technology? Well, the issues that have been raised would include that genetic modification could affect the species in many unexpected ways. This is a new technology. We don't really know all the different aspects of the genome that can be affected by gene drive technology. Also, the genetic modification is self-sustaining and therefore could spread beyond the target population or species because gene drives are exactly designed to spread. And so therefore, gene drive organisms may be able to cross international borders, and that may be difficult to actually look at, maybe because if it does cross international borders, you can infringe on the state's sovereign rights for self-determination on whether or not they want this technology. There would be a massive loss of opportunity to do risk assessment and management on their own, or it may actually compromise the state's protection goal. Gene drive organisms can affect ecosystems. And so some of the restoration steps that can usually be applied for standard genetically modified organisms may be more difficult for the products of this type of technology. Now, the landscape of governance mechanisms covers the whole range of work or activities involving the gene-derived organisms. You can start at the institutional levels, and then you can go beyond the institutional level to look at national regulatory systems as well as local and tribal levels, and especially the special concerns of indigenous peoples. And then you can look at the global governance that exists right now for technologies like gene drives. At the institutional level, we'll be looking at policies and practices that are done within an institution. Usually, this is best practiced by the Institutional Biosafety Committee that tries to prescribe the best practices that should be followed by researchers. And essentially what they want to put in place and assure everyone is that they do adhere to research standards, guidelines, and existing regulations, whatever this may be at present time. They look closely at personal qualifications, the professional standing, as well as commitment and ethical considerations of the researcher. And they ensure that adequate physical, administrative, and procedural infrastructure is available for any institution, for their institution that does gene-derived research. Additionally, we would like to have an institutional governance that will put safeguards in place in case of accidental releases, disruptions, and breach of containment. And one added, shall we say, feature of institutional governance would be transparency. The community that would be most affected by the gene technology should actually be aware of what research is going on in institutions that carry out gene-derived research. National governance is also another pathway that can be followed when you're looking at gene-derived organisms. As of now, there's no regulatory pathway that's specific for gene-derived organisms. So many of the countries that would like to look at this technology and perhaps apply it for their own research or even wider scale application tend to look towards existing governance in more advanced countries and also seek international guidance in many different fora. Most would consider that you can include gene-derived organisms in existing governance pathways. And I think this is a very, very, shall we say, good way to go. So sometimes gene-derived regulation or governance can be included in the standard regulation for pests, for drugs, pest control agents, and phytosanitary regulations, or in specific regulations for genetically modified organisms. Of course, you may also input into the governance local restrictions and concerns, tribal laws, as well as specific protection goals of particular populations. So in terms of national governance, we have some references that we can utilize. The EU law has been used as a reference by many states. It is regulated by numerous existing provisions, including those that include provisions on endangered species, protection of habitat, traditional GMOs, toxins, as well as intellectual property regulations. EU states are obligated to take steps to prevent harm from activities arising within their jurisdiction. And this is embedded in Directive 2001-18-EC, the central directive regarding GMOs, also sometimes known as the Release Directive. The US has no specific laws so they have a joint regulatory activity with FDA, APHIS, EPA, and for research, the National Institutes of Health and the National Academy of Sciences, which can provide guidance. We have specific laws also for Australia, only for genetically modified organisms or for gene technology. So that's under the Gene Technology Act. It simply considers gene-derived organisms a higher risk as compared to the classical GMOs because it is self-perpetuating. In India, there's no specific law, so they essentially just apply whatever has been created previously in their original GMO law. The African Union has decided to ban themselves, and they placed gene-derived regulation under the biotechnological framework of the Commission on Economic Community of West African States. Okay, so essentially they're looking largely at the malaria control of malaria control using population suppression and population replacement by specific mosquitoes resistant to the malaria parasite. If you look closely, all of this national governance would look at risk analysis, management, regulatory systems, and research and development that would actually be discussed and be inputted in the regulatory approaches that may be carried out in specific countries. So aside from national governance, you can have global governance because it is important that you have to look at the global issues because there are differences in national approaches to governance that may create gaps in the protection of human health and environment. So the difference in national approaches can actually be a disincentive to research, especially if the requirements are different from country to country or where the requirements are still unclear. Responsible governance has been said to include clear mechanisms for international dialogue. And these types of global governance can include formal and informal agreements, as what we have when you have guidance documents, or they can be in the form of treaties which are binding and are mandatory for compliance. So we have the major agency or the major treaty that governs gene drive as well as other gene technologies is the Convention on Biological Diversity. It's the main international regulatory instrument on the development and use of genetically modified organisms. It is an official multilateral treaty with a lot of signatories and ratifiers to this particular convention. The objectives, of course, is biological diversity, sustainable use, and fair and equitable sharing of the benefits arising out of the utilization of genetic resources. So 3 major objectives of the CBD. The CBD requires the parties to establish or maintain means to regulate, manage, or control the risk associated with the use of living modified organisms. And all the policies embedded in the CBD and all the requirements essentially would be implemented to the Cartagena Protocol on Biosafety, the Supplementary Protocol on Liability and Redress, and the Nagoya Protocol on Access and Benefit Sharing. And all this would have to be inputted to the global governance of gene drive organisms. CBD on gene drive has started, discussions on gene drive has started as early as 2010. And the discussions evolved such that now, in the last decision of 2018, the CBD has called for a precautionary approach and conditions that would limit the release of gene-drive organisms into the environment. Prior to release, it is mandated that risk assessment and risk management should be in place and public involvement should be included in the decision-making process. In the CBD and in the Cartagena Protocol on Biosafety, there is still no specific guidance document that they have issued, although talks are ongoing and discussions are ongoing on whether or not a specific document for the risk assessment of gene-drive organisms must be crafted prior to actual larger-scale implementation of research activities. The other international agreement that can be utilized would be the Convention on International Trade in Endangered Species of Wild Flora and Fauna, which aims to protect the survival of any listed species for being further threatened by international trade. Now, since gene-drive species are not likely to be part of the list, I think this convention would have minimal input in terms of global governance of gene-drive organisms. The International Bioweapons and Environment Weapons Law, embedded by the UN Biological Weapons Convention, requires countries to prohibit bioweapons. Once upon a time, this was largely confined to research on disease-causing organisms like microbes and viruses, but now it has expanded its scope to look closely at those that will limit or that would disrupt agricultural production, as well as resculpt the ecological profile. The Environmental Modification Convention prohibits hostile or military use of environmental modification and can augment the provisions of the UN Biological Weapons Convention. The IUCN is a hybrid intergovernmental and non-governmental organization. In the recent resolution, they initiated a process towards developing guidance for synthetic biology as well as gene drives. And the outcome is expected to be presented at the IUCN's next Congress scheduled this year. The World Health Organization has assigned the Vector Control Advisory Group as the major body to advise on gene drive organisms because they largely concentrate on mosquitoes. The input is critical for moving gene drive mosquitoes into the field trials. They currently support gene drive technology, encourage further development, and now requires extensive cage trial testing to be able to get enough evidence to support a field trial release. And at the way where it is going, it's going to incorporate the standard GMO regulations in looking at gene drive systems. The FAO is starting to look closely at the gene drive. They already included gene drive on the revised list of proposed thematic studies, and that they have articulated concerns that gene-edited gene drive plants can actually change the profile of the entire population as well as the entire ecosystem. And they express concern that limited policies or global mechanisms exist for the regulation of this particular technology. So what are the desirable features of governance, effective governance? Well, from looking at all these different segments or different sectors that look at regulations, apparently the most important features would include a thorough risk assessment that would involve gene-derived expertise, mechanisms for engagement of the public, delineation of authority and responsibility, balance between oversight, as well as the level of risk and adaptability of science and flexibility to social developments. My parting shot, should there be a specific governance for gene drive, or could this development be addressed by existing laws, guidance documents, and standards? We are still evolving regulation of gene drives. The concern is that if you create a specific governance path for gene drive technology, you may be setting a precedent that each novel technology will consistently require unique governance strategies. This may be too onerous to meet for certain agencies which are resource poor. It may require total dedication of new resources, and it may be, sadly, a huge disincentive to research. So thank you very much, and I'd be looking forward to the discussion that will follow this presentation. Thank you.
Thank you, Florida, for giving us a snapshot of the regulatory and policy contexts that govern the gene drive research at the global level or are in the pipeline. And we will move on to our second speaker now, Dr. Andy Shefford. Dr. Shefford, I would request him to give us an idea of the range of guidance materials and tools available to help the scientists and other stakeholders to ensure that the gene drive research is done safely and in a responsible manner. Over to you, Dr. Shefford.
Thank you, Viva. So we heard lots from Florida around the governance side of gene drives. Obviously, in order to really understand the risks and the benefits and the opportunities, we need to have effective research programs to really understand the potential for these technologies to help us. So my talk really covers on what is there around available guidance to support responsible research around gene drives, given all the pests and weeds and diseases that we're surrounded by that this new CRISPR-based technology could help us address. So I'm going to briefly cover off some introductory remarks around gene drives and their global concerns. Then I'll get into the international scientific guidance and guidelines to address those concerns, a bit on public consultation, a couple of slides on how we're going to consider building safer gene drive strategies, and then my take-home messages. So here we're concerned with self-sustaining meiotic gene drive systems. So these are genetic mechanisms, natural or synthetic, that propagate modified genes through a target population via super Mendelian inheritance. And as Florida has said, there are many of these systems that already exist in the natural world that bias inheritance. Many of them come with fitness costs, but there are many mechanisms out there that we can use to understand how these processes work in natural systems. But we're particularly concerned today around the synthetic gene drive system, which really was created as an idea in about 2002. The discovery of CRISPR-Cas9 gene shares in 2009 made it a reality, which was really firstly presented in 2014. And then from 2016, we've had all of these issues around public acceptability and ethics questions and regulations for use that have come to the fore. But let's not forget the unique opportunity that gene drives potentially provide, and that is a means of controlling pests, weeds, and diseases for which there are no other existing mechanisms. So the first case, of course, for the application of gene drives is around mosquito control. And Naima is going to be talking about that in her talk. The system that my team and I'm involved in is around targeting rodents, particularly rodents on offshore islands, to manage the biodiversity impacts that they have on threatened species on those islands. And Paul Thomas gave a talk, I think, in the first of these sessions around some of the gene drive technologies that him and his team and working with us in Australia are trying to develop. But I think just to capture it, I think there are two viewpoints of view around gene drives. One is the extreme positive and one is the extreme negative. And we need to find a balance between that. So here you've got the really excited view of gene drives, that they could be everything as an ideal control tool. They're humane, they're species specific, they're self-disseminating, they're not contagious, so they're spread by sexual reproduction. They don't require the repeated release of many animals. Whereas if on the panic side, they're considered to be uncontrollable, irresponsible, GM, won't work anyway, regulatory nightmare, international implications, ecology and trade impacts, and humans playing God. So we've got these two extreme positions, which we need to find a pathway between to be able to effectively consider the application of this technology. And of course, there have been a number of papers studied, published around the risks and the concerns around this technology, and a call for urgent review of the use of the technology. And there have been ongoing activities through the CBD and the IUCN to try and limit the development of this technology because of these concerns. But these attempts to put a moratorium on R&D have been largely unsuccessful. And of course, the CBD, as Florida was saying, is also heavily involved in considering what the opportunities and requirements are here. So the bulk of my talk really is around the international scientific guidance and guidelines to address these concerns. So what are they? And they're really covered off on into these four categories of networks and forums, of which this is one: principles, guidelines, and core commitments; peer-reviewed studies that have looked at the technology and evaluated what we need to consider in the context of being able to work on it in an ethically and publicly acceptable manner; and finally, and most recently, the codes of ethics. So I'm just going to go through each of the ones that is already available, and you'll see through these there's a huge amount of replication, but the general patterns and the general take-home messages are very similar. So in order to proceed with a new technology like gene drive, clearly we need to proceed with caution. We need the science, we need to be able to do the risk assessment, and we need the social license through liaison to be able to have developed the frameworks that underpin the science. So we've got sensible balance in the way we deliver it. And of course, in the first instance, you need to put a lot of emphasis into that right-hand end around social license and public engagement. So as I say, we've got the forums like this one, an excellent forum that's able to get these issues out into the public domain and have public discussions around them. There's a gene drive forum that's supported by the Foundation for National Institutes of Health in the US. And this forum is aimed to promulgate best practice, encourage responsible research and define what that is, and also ensure that we have safe conduct in the manner with which we undertake potential gene drive research. Very early on in the piece, this is back in 2017, a number of lead agencies around the world who work on gene drive technology published this paper in Science on the principles of gene drive research. And in summary, the guiding principles were: the activity needs to have strong public good and societal value. It needs to be supported by good governance to maintain public trust. It needs to respect transparency and accountability around the research. It needs strong stakeholder community engagement, and it needs to foster global best practice through science and education. The American National Academy of Sciences put out a review in 2016 on advancing science and navigating uncertainty around gene drives. And they came up with, in their report, with a few guiding principles for the sponsors of gene drive research. We need to advance quality science, promote the public good. We need to promote stewardship and good governance. We need to demonstrate transparency and accountability. We need to engage thoughtfully with community stakeholders and publics. And we need to foster opportunities to strengthen capacity and education. Again, extremely similar themes. The Australian Academy of Sciences did a similar review in 2017 as Australian agencies were considering this technology. Their recommendations can be summarized around communication and governance, quality and duration of science, stringent containment, risk assessment, communication and consultation, and consideration of wider implications. Recently, last year, this paper was published in Science around what the core commitments would need to be around field trials for gene-drive organisms. And again, in summary, they recommended community partnerships and good data transparency. They recommended the measuring of efficacy and safety of the technologies as they're being developed. They recommended the importance of understanding risks and benefits of the technology and working very closely with regulators as those potential field trials are rolled out. And they emphasized the importance of monitoring and evaluation of effectiveness and being able to mitigate any undesirable outcomes effectively as part of those trials. There have been assessments that have supported the understanding around the science and technology development of synthetic biology and biodiversity conservation that have also considered gene drives. And this was the one commissioned by the IUCN and published in 2019 around genetic frontiers for conservation. And it goes into great detail on the support for and the context for which research should be undertaken to develop gene drive types of approaches in a safe and effective way. And also we have gene drive roadmap, so which focused around, that was published in 2018 around risk governance and ethics, focused around this cyclical approach of problem formulation, risk assessment, risk management, and risk communication. And most recently published this year, a paper in the CRISPR journal around the Code of Ethics for Gene Drive Research, where the Code of Ethics is around conducting transparent gene drive research has to be consistent with societal needs respecting human rights, public safety, and ecological stewardship. And under scientific responsibility, the code focuses very much on the risk of research misappropriation, the prevention of the development, production, and acquisition of biological agents or toxins. We're seeing this kind of debate happening in the US at the moment around the gain of function research that's happening in that is being criticized in the context of the COVID virus. They talk about continuously assess the risks and fully timely disclosures of risk factors for society and the environment. And around ecological stewardship, you need to identify and minimize and justify any adverse effects on the public's health and natural environment. And from a public engagement and benefit sharing point of view, integrating ecological risk assessment to inform decision-making for any proposed field test, and proactively include wide-ranging discussions with relevant stakeholder communities using things like scenarios, unforeseen risks, containment and reversal options, and effectiveness likelihood. And I think all of these slides illustrate that a lot of the thinking is very well aligned, bringing up a lot of the same issues. And there's been a lot of effort put into putting in place processes and guidelines and roadmaps to assist the research and development of these technologies in a safe and equitable and socially acceptable way. Now, our next speaker is going to talk a lot more about public consultation, so I'll be very brief here. But just wanted to cover off on a couple of pieces of work that happened in the context of the consortium I'm involved in around understanding how public concerns to gene technologies. This is a paper published this year by a team in CSIRO looking at the typologies of intrinsic societal claims around human interventionist approaches. And they list out all of the typologies for those societal claims, and starting with intrinsic concerns, and then identify which ones where there is some science merit for consideration in their evaluation around. And here they've identified them as human fallibility, power and responsible use, unintended consequences, potential disastrous consequences, and reviewing previous disasters as being the core typologies where a science focus can support and help the societal considerations around the development of these kind of technologies. The team in the US, led by Jason Delborn, has done some work on trying to understand what the US public would want to know in the context of developing and implementing a gene drive technology. And here the questions they ask are ranked from those that are perceived to be the most important through those to the perceived to be the least important. And obviously the most important ones are the high-level issues around human health effects and environmental consequences of removing the pests, secondary pest potential of a gene drive system, horizontal gene transfer, and the ones that they're less concerned about are, what's the ability to reverse genetic changes and the speed of gene drive spread. So we've got some fairly detailed information, and I know they covered a range of stakeholder communities to understand where the public concerns are, at least in the context of the US. Now, very briefly going into this issue of how do we make gene drive development safer? Obviously, the classic, inverted commas, uncontrollable gene drive, which is the vehicle of most concern as the first concept of how gene drive might operate, is something that is highly unlikely to be acceptable anywhere. And therefore, as gene drive and more broadly genetic-based pest management technologies evolve, they are evolving down trying to understand and develop strategies to develop safer gene drive systems. The consortium that I work in is the Genetic Biocontrol of Invasive Rodents Consortium called GBIRD, in which a number of research partners from around the world are working collectively on a lot of these issues around the genetics and modeling, around the stakeholder engagement and field trial site assessment, around risk assessment and regulatory policy and IP, to try and work out what are the effective ways to be able to develop this technology with the safest potential, the maximizing the benefits while minimizing the risks. Some work that's under development in my own organization is the development of a gene drive utility and risk determination pipeline, an in silico process to evaluate the feasibility and risk of gene drive options against target and non-target populations and species based on population genetic data. And here you collect information to assess the populations, the genetic diversity in species populations. You identify all possible targets for genetic intervention strategies amongst the species you might want to be target. You can estimate off-target risk and then develop genetic models to study in-field propagation and use ecological models to determine feasibility and risk. So a process that's really thinking at the core about how we do this in a responsible manner to both identify the best, the most effective and likely efficacious options, while also considering all of the risks. So my take-home messages are that the potential for gene drive control, strategies are diversifying based on really on a precautionary approach recognized by all the research and development institutions that are working together around the world on this. So we're moving rapidly away from the widely criticized uncontrollable gene drive approach to looking at other options and methods to be able to limit and control a gene drive approach. As none of the institutions that are involved in this kind of research recognize that an uncontrollable gene drive approach will be acceptable. So we're moving away from that and considering a precautionary approach. The second is that the majority of relevant government agencies and reputable research and development providers, and many NGOs have signed up to these collectively agreed guidelines for undertaking open and transparent research supported by national academies and regulators. So making sure we're working with the regulators as the research is undertaken in order to address head on the governance issues that Florida talked about in the previous talk. Mostly we need to recognize that these technologies are still at the very early stage of research without field-ready systems. And so we've got plenty of time to develop this in a sustainable way. And then finally, that the research community is working on, such genetic control solutions is putting priority and equal resourcing into the independent public and stakeholder community engagement research that's now so widely recognized as critically important for the development of any new technology, including this one, to objectively address societal views and concerns, ensuring each jurisdiction can consider the application of the technology based on its unique values and ethical perspectives. So I think I can argue from my talk that there is plenty of available guidance for responsible research. Just like to acknowledge all of the staff who we work with who've helped in developing this presentation. Thank you very much.
Thank you, Dr. Andy Shepherd, for a very, very interesting presentation and making us all understand the scope of publications, experience, expertise, and all the information which is available by very, very reputed organizations who are working in this area, the science academies and the other very important stakeholders that the researchers can draw upon. These resources are so useful that they can be drawn, the research can draw upon these to inform the best practices and shape the parameters for the ethical research. Now I would like to turn on to Ms. Seitz, Naima Seitz, for an idea of how the active stakeholder engagement and public consultation fits in with these practices and governance environments. Both our earlier speakers also stressed on the need for public consultation, and now we will move on to hearing more details on this topic. Over to Ms. Seitz.
Thank you, Viva. Can you confirm, please, that you can see my slides as they're meant to be seen?
Yes, we can. We can see.
Yes, great. Well, thank you all. And I'm actually really glad to, yeah, great, thank you. I'm very glad to be going last because I get to benefit from Andy and Florida's wonderful presentations that do highlight the fact that there is ample guidance available to shape some of the research that is going on in GeneDrive. And what I'd like to highlight in my slides is the importance of stakeholder engagement and public consultation in responsible and ethical GeneDrive research. And what I'm hoping to demonstrate is that there is existing international best practice that provides some guidance. And what we hope is that while there are still gaps and challenges, identify strategic possibilities in terms of how to make sure that this is inclusive, transparent, and co-developed. So if we start with a definition of GeneDrive, this is something IFC, which is a World Bank institution on good practice for emerging markets. And this handbook provides a comprehensive overview of good practice and stakeholder engagement. However, of course, the guidance is applicable to many fields, many scales, to be selective in determining which approaches make the most sense for them. And when we're looking at some of the relevance to GeneDrive research, the key priorities I think that this handbook points out is get in early. Relationship building takes time. Many of the hallmarks of good relationships, such as trust, mutual respect, and understanding, are intangibles that develop over time. And therefore, this guidance encourages the readers and the users to consider engagement strategies that are in line with the needs of their respective projects. Another key element of the handbook is that projects, researchers should take a long-term view. Establishing and maintaining good relationships requires a long-term view. And those who benefit from this perspective tend to make different types of decisions. They invest in hiring and training community liaison staff, social scientists, and stakeholder engagement practitioners, and they see the value of this skill set and this knowledge base. They invest in translating information, not just in terms of different languages, but also taking the technical into the colloquial so that it is understandable by a broader range of stakeholders. And lastly, there's an importance in tailoring the process to fit the context. there is no one size fits all approach when it comes to stakeholder engagement. So even for us in Gene Drive, it's important that we prepare for the fact that we are entering pre-existing yet dynamic and valid contexts in the countries in which we operate, where there are established histories, cultures, knowledge, different types of expertise that are, and also often complex and nuanced political, social, and economic realities. In terms of Gene Drive research and guidance around vector control, the WHO defined engagement as practices undertaken to inform stakeholders about the disease and vectors of interest and goals of a proposed research study or intervention trial, and to understand their perspectives and reaction. The primary audience for the ethics and vector-borne disease guidance that has been issued by the World Bank is those working directly in vector-borne disease prevention and control, such as Gene Drive for genetically modified mosquitoes for malaria, including managers, researchers, and field workers. This document is a sort of a brief technical background that benefits those, even those without the scientific expertise, which I think is important, again, to making sure that there are different types of expertise and knowledge. The guidance does not and cannot really offer universal applicable answers to the complex ethical issues, nor can it provide a sort of standard checklist of issues that are necessary in all situations. However, what I'm trying to communicate here is that the goal is to help researchers recognize aspects of their work that raise significant ethical challenges and respond to these challenges in accordance with international values, best practice, and norms. When we think about, so what makes engagement for Gene Drive different, or Gene Drive research different from other types of stakeholder engagement for maybe a mining company or an infrastructure developer? The key here, I think, is to understand that there are various theoretical guidance that is very excellent in demonstrating the importance of stakeholder engagement. However, when it comes to Gene Drive research, there's still some gaps in terms of who to engage and when, considering the potential spreads. Which modalities to use, how to make decisions that produce the most effective engagement strategy to achieve responsible stakeholder engagement that places the future beneficiaries at the center of this process. To give a quick example, I'll use the Target Malaria case study. As many of you know, Target Malaria, as a project working on Gene Drive research for malaria control, Target Malaria's mission is to develop and share new, cost-effective, and sustainable genetic technologies to modify mosquitoes and reduce malaria transmission. This technology is currently at the very early stages, and it will be a number of years before any release. As Andy pointed out, there is still time to make sure that we need the research to make sure that these technologies are safe, they're efficacious, that we have a clear understanding of the risks and benefits. And that's why it's important to have the opportunity to do these investigations. And this is a sort of infographic that I hope demonstrates the importance of stakeholder engagement at Target Malaria. We have 3 key pillars, and stakeholder engagement is one of them, and pillar. And for us as a project, we've identified very early that co-development is a key value. Co-development, in our definition, is a collaborative process of jointly designing a research pathway and its resultants to reach a common goal. And in Target Malaria, what we've noticed is that as an engagement strategy, it needs to be iterative. It needs to take into consideration a deep understanding of the context. It has to mirror the project's principles and values of openness, excellence, transparency, inclusivity, and needs to have the mission of malaria control at its center. However, we really do benefit from various recommendations and guidance from the WHO, from the National Academy of Sciences, from different papers that have been written by various experts. And there is also the recognition that the context will change and that feedback from stakeholders will change, and it needs to be integrated into our approach. And that is why, as a stakeholder engagement team, we have at the center of our activities the national level, local, in-country social scientists, communications experts, and stakeholder engagement practitioners who really drive the implementation of this strategy on the ground. There are still challenges, and this is where I think it's important to recognize that even as stakeholder engagement practitioners, we too are researchers in the field. We also have issues and challenges that we're seeking to investigate further. And it's important to see that there's still some challenges around how do you really define and identify the key stakeholders in gene drive, considering that there is the potential for spread. It's important to find the right balance between engaging proactively and in a timely manner, but also communications being different from other types of communication when you're also committed to being open, but with a level of uncertainty around how the research is developing. And I think it's just another point to make that we want to make sure that stakeholders understand that gene drive for malaria control is not a silver bullet, and that elimination and eradication of malaria will require a full and extensive toolbox of both existing tools and other new tools, such as the vaccine and others in the future. Just to touch upon public consultation. And as the previous panelists also mentioned, the Convention on Biological Diversity is a critical piece in public consultation as well. And the conservation's priorities of conserving biological diversity, the sustainable use of components of biological diversity, and the fair and equitable sharing of the benefits arising out of the utilization of genetic resources is something that also prioritizes Indigenous peoples and local communities, and their understanding and informed consent agreement of the technology and of the release are critical. And also, as mentioned by Florida, the Cartagena Protocol on Biosafety to the Convention of Biological Diversity is an international agreement which aims to ensure the safe handling, transport, and use of living modified organisms resulting from modern biotechnology that may have adverse effect on biological diversity, taking into account the risks of human health. And even in the Cartagena Protocol, there is a clear highlight of the importance of public consultation and making sure that the public and key stakeholders are aware and informed, and that information is accessible to all those who are interested. One point that I would like to raise, though, to really distinguish here is the difference between the roles of the governments and the roles of the developers in public consultation. And when it comes to stakeholder engagement and public consultations, there are established ways of working at national, regional level that are separate from the role of the development and the researcher. However, it's important that these two are recognized to be interdependent and that they work together in achieving the common goal. Existing guidance in public consultation is, as I mentioned, informed by various literature from the National Academies of Sciences, Engineering, and Medicine, as well as the WHO, to name a few. And in terms of the guidance framework that the WHO has recently updated for genetically modified mosquitoes specifically, this guidance framework is intended to foster the quality and consistency of procedures for testing modified mosquitoes, which will contribute to the comparability of results and credibility of conclusions to support decision-making. This guidance framework is also very useful for researchers, but also generally readers interested in genetically modified mosquitoes technologies and the applications that are currently being tested. or contemplated, safety, efficacy, regulatory, and social ethical issues involved in genetically modified mosquitoes, as well as existing regulatory frameworks and international agreements relevant to the testing and eventual potential implementation of genetically modified mosquitoes. An example here is from, again, Target Malaria. In 2020, and there was a workshop by Target Malaria, the Kenya Medical Research Institute, and the Pan-African Mosquito Control Association, which co-organized a workshop with researchers and practitioners from Africa, Europe, and North America on the topic of a potential model to be proposed for the release of genetically modified mosquitoes in the future, and how future studies on gene-drive mosquito releases could offer reflections and best practice. This paper shares the outcome of that workshop and highlights the remaining topic for discussions needed for a comprehensive model to be designed. And lastly, thank you. And I just wanted to state once again that stakeholder engagement and public consultation are critical aspects of responsible and ethical gene-drive research. And while there is existing best practice that we benefit from on a day-to-day basis, there still remain some gaps and challenges in this field of area-wide vector control, including but not limited to gene-drive. And I truly do believe that the key to answering these gaps is an inclusive, transparent, and co-developed approach towards these challenges. And it is needed to make sure that an agreement model for potential gene-drive releases is considered to be and actually is legitimate for and by key stakeholders. So thank you very much.
Thank you, Ms. Sykes, for a very nice presentation and explaining about how the stakeholders' needs and perspectives can be integrated into the research pathways, and the importance of involving the stakeholders at every stage of the research. This is very, very important, and the examples given by you from the Target Malaria experience were also very useful. So I once again thank all the three speakers for giving us a very good overview of the topic of today, which is the regulatory and governance considerations. We heard about the global initiatives which are there, the country-specific regulations, and then moving on to the available guidance for the responsible research and the stakeholder engagement. So we have some time, about 20 minutes for the discussion. And so I will just read out some questions which are there in the chat box from the audience. And then maybe we can move on to some other issues like the need for harmonized regulations, on which I would also like the views of the speakers. So I will just go through. There are a few questions. Dr. Florida, there is one question for you that based on the various regulations in the diverse regions that you have presented, in your opinion, are they effective and sufficient in addressing the gene drive, or they need, your views on that? He has actually responded on the thing, and so maybe I will take his views also later. So Florida first.
Yeah, I think it would depend first on the gene drive organism that you wish to deploy, or the activity, the level of activity that you wish to engage in. I don't think any national or international regulatory body will be able to come up with a perfect solution. It would all depend on, one, what are your protection goals? What is the organism being introduced? And what is it supposed to be doing? And so you can mix and match certain provisions of different regulatory pathways. And I think the flexibility may allow you to come up with an assessment and a pathway that will at least assure some level of safety and allow the mitigation of risks that may be associated with the gene drive activity that you wish to undertake. So the answer is, I don't think anything is really a perfect match for any regulatory system that you wish to deploy in a given environment. So I think the regulatory system has to be flexible enough to be able to accommodate specific entries that you wish to work with.
Yes. So that's very correct. There are additional considerations for gene drives, and they need, of course, some more levels of assessment probably. But in the broader framework, the research and other activities have been initiated. And for the particularly maybe moving forward for release, etc., there would be additional considerations. Andy, would you like to add something to what Florida said?
Well, I think I just want to reiterate what Farida said in her talk, which is that we don't want to be moving down a path where we develop unique regulatory mechanisms for every single new technology that comes down the pipeline, right? So the best approach is to have an effective regulatory system around genetically modified organisms, and modify that over time as the technology to keep up to speed with how technology is changing in that space. So that's what we did in Australia. We sat down and talked with and worked with the regulators from the moment the opportunities around gene drives became apparent. And we discussed with them, help them learn the changes in the technology to understand how they should modify the existing regulations so that they would be continue to be effective to evaluate these kind of technologies.
Yeah. Another question to you, Andy, that there's a question from the participants that whether the less developed countries can really control this new biotechnology. So, I mean, the kind of regulations they would require, or how they can, manage this kind of a research, because there are so many apprehensions and concerns in probably people's minds. So, would you like to respond to this?
Well, I'll make a brief comment, but then I'll pass to Farida because I'm sure her expertise is better than mine in this space. Because I don't work in lesser developed countries, So, but as I say, this I'm sure has been a challenge for those countries since the first GMOs were developed, right? So again, it's around how are those countries' regulations addressing GMOs? If they're not, then clearly they need support from other countries in being able to develop that kind of regulation. But that regulation needs to be kept up to date in the context of how technologies are evolving. But I'm sure Florida's got a better answer than me.
Yeah.
Well, let's put it this way. I think the developing countries who are able to work with ordinary or shall we say standard GMOs would be able to adopt the regulatory system to be able to accommodate the gene drive organisms. They would have to put in, of course, information on what they already know about the unmodified organism. Look at population biology of the organism that they wish to do the gene drive experiments on. And then proceed on a step-by-step manner. In my country, we're lucky we are an island ecosystem. We have several islands which you can really use for limited releases and effect, for example, a temporal and a spatial containment. But other countries, I think, would be able to do that, provided they are able to open their minds on the various levels of engagement with gene drive organisms. Proceed step by step, relatively slowly, know more about the organism, try to project what its adverse consequences would be, the introduction of the effect of this on the receiving environment, and then weigh the options. The risk-benefit analysis would come into play. And I think if they are able to at least outline the adverse consequences of the deployment of these organisms, they would be able to manage the risk associated with it. So I think existing guidance documents for regular GMOs can be adopted towards this. Plus, of course, the existing information and guidance for invasive species can also be put into play, as well as control of insect populations, for example. Those are information that can be inputted towards a regulation of gene-edited organisms. Yeah, mix and match, I think, would be able to help us control these organisms.
Yes, very right. I think it's not a question just of the developed countries doing it, but definitely, like Florida said, the less developed countries can also develop the management systems, the regulatory considerations based on the experiences, and can definitely, take benefit of all these technologies in their countries.
Viva, if I may,
Hello.
Sorry, I just wanted to add a bit also from the African perspective.
Yes.
And that is to say that I completely agree with Andy and Florida's points that even in West Africa specifically, but in generally the African officials with the support from the African Union Development Agency, AUDA and NEPAD, have already started thinking through these questions and have been preparing joint guidelines on how to evaluate any potential request for work on gene-drive organisms and the creation of a regional platform to manage these vector control tools. And I think that for Florida is definitely much more well-versed, but my understanding of the CBD and Cartagena protocol, the implementation of some of these key points is to make sure that there is fair and equitable resource and guidance available to both developed and developing countries. And so I think it's important to recognize here that in the space of gene drive research, there is the regulation in place and the governance in place to make sure that sort of no country is left behind that is interested in gene drive. The tools are there to make sure that the guidelines are followed, to make sure that regulators are given the support they need to follow through. Thank you.
Thank you, Naima, for sharing your experience. I have, there's one question, you know, although you mentioned in your presentation that all such organisms are far away from release, but still we have a question that can you please list some gene drive organisms in use now? And also, you know, if you could kindly share, you know, like an overview of research, the stages where we are in terms of, you know, regulating or in terms of research of these organisms. Can you quickly mention if you have information, Naima?
Oh, well, I can only speak on behalf of Target Malaria, and I can say that there is currently absolutely no gene drive mosquito flying anywhere. And so for us, the key is to say that we are very, very far from release because of everything we're talking about today, that we follow a stepwise approach in our development pathway to ensuring that all risks are measured, understood, before decisions are made. And it's important to recognize that in the development pathway for genetically modified mosquitoes, there are different aspects that must be taken into consideration from the lab to release. And this also means that there are non-gene drive sterile male mosquitoes that have been developed. But again, those are non-gene drive. They do not procreate. And so this is all part of the step-by-step approach. And it's just to also assure, I think, any concerns that the decision to release will be made by the key stakeholders in the regulatory policy space in each of these countries and each of these regions. That is not a decision that will be made by the developer.
And if I could just follow up more broadly, I mean, the concept of gene drive is more developed in mosquitoes than in any other system. So that assures you that there are no gene drives anywhere else either. In fact, getting a functional gene drive in a vertebrate has been a real challenge. The system that's been developed in mosquitoes does not work in vertebrates. And so people are coming up with other approaches that may not in fact end up being a gene drive to be able to manage vertebrate pests.
Yes, thank you, Andy. And continuing with that, you know, there's another question from the participants that to what extent can the experiences with rodents and insects be extrapolated to, for example, plants and fish? So I think the research is underway, but at this point of time, as you said, you know, the genetically modified mosquitoes, the gene drives in mosquitoes are at the most advanced stage. So maybe you would like to add something here?
Yeah, look, I can add something very briefly. So we are doing some work in Australia on a fish system, but again, it's another vertebrate, so it's hard to know exactly how it'll pan out. But we've started some research on looking at whether or not we can develop a gene drive type working system in a fish. And there's certainly a lot of thinking going on about whether they can work in plants. Obviously, those plants have to be sexually reproducing plants, right? But plants are complicated organisms with very large genomes. And so I think plants are recognized as being, you know, probably a harder one to try and tackle. So we're focusing primarily in the context of the work we're doing on vertebrates.
Thank you. Thank you, Andy. And I will have one question to Florida, and to Andy, to all the three panelists, that we've seen that, we all said that there are regulatory frameworks in place, as Naima also emphasized, that in various countries, and those frameworks can be used to initiate the research. And that coupled with, all the resources which Andy mentioned, the guidance by the WHO, by various academies and all that, it provides for a very, very useful start, for having a harmonized guidance globally. So just your views on, that what about like having a uniform framework? Because gene drives are definitely, there are concerns in the minds of people that they are different from GMOs. There are some additional considerations. So just your views on, general views on how, how we can move towards a harmonized set of regulations for dealing with all these considerations.
Well, my view is that probably needs to happen in the context of the international conventions and organizations that are considering this, to be able to come up through their negotiations with discussions on how you could have an overarching framework to support this. But I just also want to say that, as I say, the classic gene drive, as it was explained in one of Florida's slides, is only one option. There are many other options out there being considered, because gene drives don't work as easily as they were originally thought to. And so I think we've got to recognize that the technology will evolve, and I'm talking broadly about genetic tools to manage pests, weeds, and diseases are going to evolve in a number of different directions. And the classic conceived gene drive system is simply one of those. So again, we've got to be cognizant that the world is changing fast, and the regulations need to keep up with the technologies.
Absolutely.
Yeah.
Absolutely.
If I were to make a comment, the problem is this. I think when you start talking about gene drive organisms, we forget the fact that these are the same organisms that we have been familiar with. And they will contain most of the biological properties of that particular species. So we have familiarity with the species, and we add on a particular trait, and that is population control. But we already have information as to the dispersal range of the organism, its mating behavior, perhaps, its ability to interact with its environment, its biological and ecological function. These are already information we have on hand on the biology of the organism that we have to which we have introduced a gene drive mechanism. So that would factor in greatly in doing a risk assessment and trying to predict the consequences of deployment. So I think we don't have to start from scratch. Neither do we actually have to come up with a single harmonized guidance document. The only thing that we need, I think, would be certain bullet points on what aspects to look at, what to include in a risk assessment, how can we predict what the consequences are. And perhaps the only important capacity building program I can think of right now is in the modeling stage. How can we model stage based on what we know and based on the efficiency of the gene drive that we have introduced into the organism? So I think that's where we must build capacity on the modeling part. Thank you.
Yeah, and I'd argue that Target Malaria is the project that's really pushed the boundaries on producing world's best practice around risk assessment. You know, the risk assessment work that's gone around Target Malaria provides you with the best possible practice you would need in order to be able to evaluate the risks of any other genetically modified organism in this context. So we've got a lot of that work. It's already been done.
Mm-hmm.
Yes, I think that is a very good point, that the best practices which are there through the management of these projects can actually provide a lot of guidance. So I'll just move on to one more question which is there from the audience. If we are genetically modifying the species and only studying the gene level for assessment of any risk, it will also include any protocol, ? So I think there are very good risk assessment, like Florida said, the risk assessment protocols, modeling, etc. But maybe one of you would like to answer this and to the applicants, this one. If we are genetically modifying the species and only studying the gene level for assessment of any risk, will it include any protocol?
I don't fully understand the question, but I would say that when we do the risk assessment, we don't just consider this to the gene level, right? We consider every aspect. In fact, we get the whole community in the room and we get them to tell us what they think are the risks and define the risks, and we evaluate them all.
Yes, thank you. There's one more question from the audience, which I would like to pose to you all with your experience in dealing with the regulatory committees and the researchers. Can you please comment on the question of capacity building that might be, required for the regulatory bodies for, taking forward this new technology? Any kind of capacity building needs that you all can, share with us, which can be taken forward by the organizers in further initiatives? Farida, any kind of...
I didn't get the question. I'm so sorry.
Capacity
I have a choppy internet connection right now. Sorry, I didn't get the full question.
No.
Well, I think, I mean, my understanding of it is, and I think it's a really, if I understand it correctly, it's a really good idea, and that is that regulatory bodies around the world should be networking and talking to each other about how they're trying to address these issues. So it's not just the scientists that are working together around the world, or the governments, but the regulators are actually working together to come up with effective regulatory processes. Because, I mean, to be honest, the regulatory process in the United States is very complicated because they have a very complicated legislative background over which they need to evaluate it. So you can overcomplicate this as well as, and the opportunity is, if you're coming into new to the game, is you can come up with something that is both effective without blocking the progress.
Yeah. Thank you, Andy. And Farida, you being so active in the regulations, could you please suggest any mechanisms, good mechanisms for ensuring that the countries are looking at, the technology, evaluating the technology in a similar manner, so that what kind of capacity building we should be done, the needs that the ultimate evaluation is in a similar fashion and not additional requirements from one agency to another, which would be difficult for the developers.
Well, I think the major point that could be shared will be information on the biology and ecology of the organism to which the gene drive has been introduced. And then we can learn from the experience of others. As I said, in most countries, we are basically up to scratch with respect to knowing the biology of indigenous organisms. But for alien species, perhaps, the information can be shared. There's a lot of biological information out there. Perhaps capacity building can include citations of these references. But as I said, in the Philippines, at least the way I say it is that we lack modeling capacities. We're not very good modelers. We have very few epidemiologists who are being roped in sometimes into coming up with distribution and genetic drifts. And of course, they don't understand too much about this. Even Hardy Weinberg is strange to them. But they would rather be modeling, for example, the disease spread and the epidemiological context of disease. So I think the part wherein capacity building would be most helpful is to be able to come up with models that would show dispersal mechanisms, dispersal rates. And I think that is at least the initial step that should be taken when countries even at classical GMOs to be able to limit their unwanted or unregulated dispersal. So yes, that's the way I see it with the present regulatory system that we have in the Philippines, as well as in some of the countries in Southeast Asia.
Right. So this means more resources and also interactions on specific, issues would be very useful. There is another question, which is of course very general, not related to regulations, but maybe I can ask Naima, with her experience with the communities. The question, of course, says that, do you think gene drives can achieve same result as intensifying efforts in the production of vaccines and diseases of medical importance? So I'm sure you would have come across many such questions when you are doing the community engagement, even in target malaria. So can you just share that, how such questions are dealt with?
Yeah.
The strategies for dealing with such comparative efficacy of, these gene drives versus the other options How
Yeah.
do we talk to people, to different stakeholders?
So I think I touched upon this briefly in my presentation, but thank you for giving me the opportunity to highlight it once again, that the nature of malaria and the malaria parasite means that you cannot have one silver bullet. So existing tools have done a lot for saving many lives for many decades, and that whether that's mosquito nets or anti-malarial drugs, they have successfully for a long time saved many lives. However, with the evolution of this disease, the fact that it continues to kill hundreds of thousands of majority Africans a year means that new tools are needed if we do want to eliminate this disease, which is such a heavy burden, not just for public health, but for society and economies and entire nations. So I think what's important here is, again, to recognize that gene drive is not to be considered a silver bullet. And that is something that we communicate to the communities. You must still continue to use bed nets. You must still continue to protect yourselves. You must still continue to finish your dose of anti-malarials if you do get malaria. And even with the discussions on vaccines, we are 100% supportive of the need for vaccines. And they too, for malaria, are still at an early stage. And we were, I think early this year, the milestone of the most recent study proving to be, I think it's 77% efficacious is wonderful. But even our partners in the malaria fight recognize that it too is still has a long way to go. And so we need as many tools as possible to fight this burden that is destroying so many lives.
Thank you so much, Naima. And I think with this, we had a good discussion and good questions from the participants, very ably answered by our speakers. I would just like to briefly conclude before giving it back to the organizers. We've had on this very interesting topic, and very, like I said in the start, this is a very, very important topic, because to take forward any new technology, you need most appropriate regulations. And this was something which was echoed by Dr. Florida Carino, that there are several levels of governance and they can be tailored to specific activities, including the gene drives. Existing guidelines, rules, treaties are flexible enough to accommodate this new emerging technology, that is the gene drives, and the governance mechanisms for that. There is creating new specific governance strategies for each new technologies can be very daunting and wasteful of resources, and this can always delay the adoption of the technologies and bringing the benefits of the technology to the society. We also had a very interesting presentation from Dr. Andy Shepard, who introduced the really useful guidelines which have come from very reputed research and development providers, NGOs, academic societies. And this is helping, that this is an early stage research, and there is enough, I would say, the guidance available to take it forward in a most ethical manner. He shared the latest resources. The research community is working on such potential solutions, and they are also putting in, as was evident from Andy's presentation, and the interest of all these academies, etc., to put forth these resources, is that the independent public and stakeholder engagement is being given to understand and take into account the views and concerns for all sectors of the society, and allowing each country to consider applications from their own social ethics perspective. Naima's presentation, very good. Thank you. And Naima, stakeholder engagement and public consultation is very, very important. And we really heard, that these are the fundamental aspects, particularly in for the responsible and the ethical gene drive research, where it is not an individual's decision to deploy a product, but it is a community product. And that is why the community engagement is all the more important. There are existing international best practices which are providing guidance to the developers, but there is continuous research, and all the gaps and challenges are being addressed through initiatives such as Target Malaria, who are actually working on the ground to bring the benefits of this technology. With these words, I would once again thank ISAAA for giving me an opportunity to moderate. This has been a very, very learning webinar, learning exercise to hear from the three esteemed speakers on the regulatory and governance considerations. I would like to now request, give it back to the organizers and request Dr. Maha from ISAAA to say a few words and close the webinar. Over to you, Maha.
Thank you so much, Dr. Viva. Thank you to all the speakers, panelists, such rich and very enriching webinar. I'm not going to summarize. Viva did a great job. I just want to thank all of you for this seminar. I also want to thank Outreach Network for Gene Drive Research for giving ISAAA the opportunity to host these webinars. And it's not just one webinar, we did a series of 4 webinars, and this is the last of the series. And we are happy to continue this if there is a need. And I'm sure there is a need because Gene Drive is extremely new technology. And from the start, when we started with the first webinar, it was on just the basics of Gene Drive. And then we went into how Gene Drive approaches support or play a role in recovering loss of biodiversity or conserving biodiversity. Then we went into gene drive approaches in vector-borne diseases. And today we discussed the governance and regulatory aspects. And we know it's not done. There's so much to do. The stakeholders still have so much of question. And like what Naima and Dr. Viva was saying, it's a community centric approach. So we need everyone to come together. So I'm sure more outreach program is needed, and that is what ISAAA does. So please feel free to approach us if you need any other information. If you need the network, we can put you to the people, and we look forward to organizing more outreach programs. Exactly, that's what ISAAA's core objective is. I would like to ask EJ, yeah, he has already put again the link to the evaluation form. Please fill in the evaluation form so that you get your digital certificate. And also one final reminder is that all the previous webinar is still on our YouTube, ISAAA YouTube. So please go to our ISAAA YouTube and you may watch if you have missed any previous webinar there. And please also share it because we want this discussion to go on. So please share it with your network. So with that, I would like to close the webinar, and thank you so much to the participants. Thank you so much to the panelists, and of course again to Isabel, Zaira, and the team for entrusting ISAAA with this huge responsibility. We hope to work together again. Thank you.
Thank you.
Thank you.
Thank you so much.